Our ears, our eyes, our hair, and our noses look the way they do because of DNA. But how does the DNA magically result in a trait? What really happens in between? Welcome to Transcription and Translation.
Transcription is the synthesis of mRNA by an enzyme called RNA polymerase and occurs in the nucleus. mRNA is like DNA, just single-stranded.
It requires DNA and free RNA nucleotides in addition to the RNA polymerase. Remember, nucleotides are the basic units of nucleic acids. They consist of a nitrogenous base, a phosphate group, and a sugar. Because RNA has A, G, C, and U, A pairs with U and C pairs with G. The RNA polymerase binds to the start of a gene and unwinds the DNA so that two separate strands of DNA are formed. On one of these strands, called a template strand, the RNA nucleotides form complementary base pairs with the DNA nucleotides. The new mRNA is released from the nucleus when the end of the gene is reached. Keep in mind that transcription only occurs for one gene. The entire DNA sequence is not being transcribed. After transcription, we move into translation. Translation of what, you may ask? Well, mRNA and DNA language is essentially being translated to amino acid and protein language, because translation is about making the proteins that result in the physical features that we have. In order for translation to take place, we need mRNA, tRNA, and a ribosome. mRNA is the messenger that sends code from the nucleus to the ribosomes. tRNA transfers amino acids, the building blocks of proteins, while using the code that the mRNA provides. Transcription happens in the nucleus, while translation occurs in the ribosome, a protein-making factory. First, the mRNA leaves the nucleus and travels throughout the cell until it binds to the ribosome. Then the first tRNA and the mRNA will bind, or more specifically, the first anticodon and the first codon. A codon is a set of 3 nucleotides on the mRNA. The bases on the codon and the anticodon are complementary. The codon corresponds to one amino acid. The first codon is typically AUG, which corresponds to the amino acid methionine. After this, the second tRNA with its amino acid arrives. The 2 amino acids are linked by a peptide bond. The first tRNA will leave, and a new tRNA with a new amino acid will arrive. Only 2 tRNA are able to bind to the mRNA at a time. As the mRNA is pulled forward, the amino acids continue to bind to one another as the tRNAs keep coming and leaving. At the last codon, which is UAA, UGA, or UAG, ta-da! We have a protein. But we're not quite done. After translation, the polypeptide will go through certain edits, and will also be folded into this structure. After that, the new protein is directed toward a certain part of the cell to perform its job. And that is how we get from DNA to protein to our physical features.